Hi, my name is Eric Adams. I'm the president and CEO at InMed Pharmaceuticals. Today, I would like to introduce to you our lead drug development candidate, INM-901, which is targeting neuroinflammatory pathways in Alzheimer's disease. InMed is a publicly listed company on the NASDAQ Stock Exchange under the symbol INM. So before we get started, it's important that I point out to you that I will be using forward-looking statements in the presentation today. A full description of these forward-looking statements and explanation is available on this slide and also available on our website. I probably don't need to tell you much about Alzheimer's disease because we've all been affected in one way or another by it. But it is a subset of dementia that impacts the part of the brain that controls thought, memory, and language. And this disease leads to increases in illness and death. It's a very big problem, not only in terms of the lives that are impacted, but society in general. It's the sixth leading cause of death for individuals over the age of 65. So up to now, a lot of the research in Alzheimer's has been focused on two specific targets in the brain. One is called amyloid beta plaques, and the other one is called tau protein tangles. And these are physiological conditions that seem to be elevated in people with Alzheimer's disease. But now the thinking has started to shift, and we're looking at other areas that may be contributors to the underlying disease, and addressing them may enable us to slow down or even potentially reverse the disease. One of those in particular is neuroinflammation. So just to give you some perspective on how the medical research has evolved, as I mentioned previously, amyloid beta and tau proteins were thought to be the root cause of Alzheimer's disease. And so the thinking was, if you can address these, if you could knock these down, then this would lead to a halting or reversal of the disease. So these amyloid beta proteins and tau proteins lead to neuronal damage, the damage of the neurons in the brain that give us thought and consciousness. And that in turn leads to an inflammatory response that it comes in to clean up all of the damage. So that's the old way that this disease has been considered. The new way looks at it a little bit differently, and that is that this is really a vicious cycle between all three components, and maybe more, but certainly these three, between amyloid beta and tau, the neuronal damage, and neuroinflammation. So the disease can actually start with any one of these that then impacts the others in this cycle. So, a lot of research has gone into the amyloid beta theory and the tau theory. We're coming at it from a different direction, and that is the neuroinflammatory portion of this cycle. So to date in the Alzheimer's space, there are two approved disease-modifying products. Both of these are large molecules, and they target amyloid beta in particular. There's over 130 drugs that are in 182 clinical trials right now. So it's a very extensive area of research, and it needs to be so, because this is such a disease with a high unmet medical need and impacts us so severely. If you look on the left-hand side here, the therapeutic targets that are being studied, you can see that neurotransmitters, amyloid beta, and neuroinflammation are kind of at the top of this list. We fall squarely into the neuroinflammatory aspect of this, but there's a lot of different approaches that are being researched in order to address this disease. The pathophysiology of Alzheimer's disease is very complex, as are a lot of diseases. And so this slide is really meant to depict some of the preclinical data that we've observed from 901 in a number of different models. So starting in the upper left, neuroprotection, what we observed is that in the presence of 901, neurons tend to live longer, not only just under their normal state, but also when they're stressed by amyloid beta plaques. So the drug is somehow acting to preserve these neurons and preventing cell death. Below that, you can see the subset neurogenesis and neuritogenesis. In this case, the neurons themselves tend to have more fingers that stretch out, and these fingers are actually longer. And I'll talk a little bit about why that's important, but it's a very interesting effect. And in the lower right-hand side, the amyloid beta plaques, we have seen a reduction in amyloid beta in the presence of 901. So we don't think that's the primary effect that's going on here. It's certainly interesting, but there's probably other drugs that do a much better job of reducing amyloid beta load. But in the upper right-hand side there, the neuroinflammation, that's really where we think we're going to play an important role. And if you look to the side there, in Alzheimer's disease, these markers, these biomarkers that can be used to measure inflammation in the neurons, all increase in the disease state with Alzheimer's. However, in the presence of 901, we can demonstrate a very marked reduction in all of these markers for neuroinflammation. So that's the anti-neuroinflammation effect that we're seeing with 901, and we think that that may play an important role in the treatment of Alzheimer's disease. After seeing such exciting data in a preclinical setting in vitro, we then moved on to conduct animal studies using a Alzheimer's disease model. So these are animals that have been genetically modified to have an increased production of amyloid beta, which then leads to a Alzheimer's-like state. If you look on the left-hand side there at the top, it talks about the experimental design, and this was a very thorough, very long-term study to look at this animal model. There's 5 boxes there that depict the different treatment groups that we had, or study groups. On the left-hand side, the wild type, these are just normal mice, and they were used as the controls. So we use them to see how normal mice act and what happens in their brain as they develop. So we had one group that we just left alone, that was the placebo group. We had another normal mouse group that we actually administered the drug to see if there's any potential negative effects of the drug itself on a normal mouse. The next 3 groups were all these diseased animals called 5XFAD mice, and there's 5 genes that are knocked out in order to lead to their ability to have advanced Alzheimer's disease or amyloidosis. The first ones were just normal genetically modified mice. That was the genetically modified placebo group. And then there were 2 groups that received INM-901 at a low dose and a high dose. These animals were started to be treated at 2 months old, and they were treated twice weekly for 7 months. So it was a very long-term study. During the course of this study, what we did is we evaluated a lot of the behaviors of these animals in a number of different tests. And I'll go into more detail about each of these, but it were really tests to see that, how does a normal mouse act? How does a diseased mouse act? And then if the diseased mice receive our drug 901, how do they act? Is there an improvement in their behaviors? And those were conducted throughout the course of the study up to 9 months old. At the end of the study, we collected brain tissue and blood, and we ran a number of different microbiological tests here to see what was going on internally with various markers for inflammation and other disease markers. So it was a very thorough, very long-term test, and we're very pleased with the results that came out of this test. We took the blood of these test subjects to really try to measure the impact of our drug on the neuroinflammatory markers in this disease. And so the way to read this, going from left to right on any of the charts, the first bar in blue is the normal mice. So if you measure their blood, for instance, for IL-1 beta, at the bottom there, you can see what the normal mouse level would be. The next bar over is the untreated diseased mice. And you can see for IL-1 beta, there was a stark increase in the amount of this inflammatory marker, meaning that there is a strong neuroinflammatory response in these animals to the disease. The next 2 bars are the diseased mice who were treated with a low dose and a high dose of INM-901. And you can see in all of these cases, first of all, there was a marked increase in the diseased animals versus a normal animal. And then when the diseased animals are treated with our drug, they tend to return back towards the baseline of the normal mice. And in just about all instances, we had a statistically significant improvement in the reduction of these neuroinflammatory markers, which first you want to see a reduction. The second thing you want to see is that a high dose works better than a low dose. So that's what we call a dose response. So two takeaways from this. The first one is that the diseased mice exhibited significantly elevated levels of these markers compared to normal mice. And secondly, when treated with INM-901, there was a significant reduction in these biomarkers, and also suggesting there was a dose-dependent therapeutic effect of INM-901 in this animal model. We also looked at behavioral tests in these animals to try and see whether or not there is first of all, an increase in these anxiety-related behaviors or the decrease in cognition due to the disease itself. So again, we compared the wild type or the normal animals with the untreated diseased animals, which is depicted here in the gray bar. And across all of these tests, what we saw is that the diseased animals had cognitive impairment, behavioral impairment, and reduced sensory responsiveness, which is what you would expect in a diseased animal. We then treated some diseased animals with our drug to see whether or not there was a reduction in this state and whether or not these animals started to return towards baseline, which we did see in all instances. And I'll talk about each of these in a different slide, but you can see in summary across the board in all of these validated tests that are standard for testing drugs in this arena, we had an improvement, and it was a dose-dependent improvement, meaning the higher dose group responded more or had a better outcome than the low dose treated groups. So overall, INM-901 demonstrated a trend in improvement in behavior across all of the tests that we used. Now, most of these were from the long-term study. The novel object recognition there in the middle with the rubber duck, that was conducted in a similarly structured and designed trial that was a short-term trial. It was only a few months versus the 7 months of the long-term one. But across the board, we saw an improvement in these behaviors. So a little bit more detail on the neuroprotective effects that we were able to demonstrate in vitro. This test, you basically take these neuronal cells and you expose them to amyloid beta, which is the first column, and then they die at a certain rate. And you take that rate and you basically set that to 100%. So in the presence of amyloid beta, this is the level of death of these neuronal cells at a certain time point. We then took cells that were exposed to amyloid beta, but were also exposed to INM-901 at different concentrations. So you can see here that as you introduce 901 and as you increase the dose, you had a dose-dependent effect in reducing the percentage of cells that died in this culture. And so it's nice to see the reduction. The last two are not statistically significantly different, so you can imagine those as being the same level. So we did see basically a plateauing at around 15 to 20 micromolars, and this tells us that the INM-901 treated arm demonstrated a dose-dependent cell protection in this model. I spoke earlier about neuritogenesis, and that was one of the effects we were able to measure in early preclinical testing. The neurites are these finger-like structures that branch off of the neurons. And in diseased patient populations, we see two things. Number one is that these neurites are shorter and that there's fewer of them. And as these neurons mature, they're supposed to extend not only the number of branches, but also the length, so that the neurons can communicate with one another, and that's what gives us our cognitive thought and memory. So it's important that you have healthy neurons, that you have very long branches, and that they don't decrease or diminish in the number of branches that you have. So what we saw from our in vitro testing was that INM-901 demonstrates increased neurite formation. It's a little tough to see here, but if you look at the images, the differentiated neuronal cells that have been impacted are taken, and you can see under a microscope and under fluorescence that they're very fragmented, they're short, and that leads to the disease state. We then took these and exposed them to INM-901, and you can start to see that there is the neurite length is longer and that there's more of them. In high magnification, you can see here the little bitty branches that are starting to form and the length of the normal healthy neurons, the neurites. So under all concentrations that were delivered, you can see a significant increase, in most cases 40 up towards 50% improvement in the length of the neurites. So this tells us that the INM-901 treated groups display extended neurite length, which signifies an improvement in neuronal function. So in summary, we are building a very significant library of data that shows that INM-901 potentially mitigates the pathology in Alzheimer's disease. There's a number of different aspects that we've been able to show: the anti-inflammatory action in animal models, a very robust long-term animal model. We've also looked at a second model called an LPS-induced inflammation model, and we've seen similar anti-inflammatory action there. We've seen neuroprotection, as we've talked about. We've seen a lot of validation of the behavioral outcomes at a molecular level. We also have this drug now formulated to be delivered orally, which we think is important, and it doesn't have to be delivered by IV. We've seen neuronal regeneration, and we've seen a significant improvement in behaviors in animals who are diseased and are treated with 901. So we continue down this path to collect the data that's necessary to advance this compound into human clinical trials, and we'll be updating everyone as we continue down that path. So where do we think this product is heading? Well, currently there are some anti-amyloid beta monoclonal antibody therapies that are available. They have some effect. I think most would agree that the effect has not been as impressive as everyone had hoped. And there's only been limited evidence of restoring lost cognitive function with these drugs. It's still yet to be tested in human trials, but we think INM-901 could potentially reverse disease effects and restore cognitive function based on the preclinical data that we have seen to date. There's a number of side effects with the current therapies that we do not anticipate seeing with 901, just based on the chemical structure and the way that the drug acts on the disease. And as I mentioned before, an oral formulation is much more preferable than an IV infusion. It's much easier to administer to the patients. So we think there's a number of scientific reasons why this drug candidate, INM-901, will play an important role in Alzheimer's disease. And of course, that has to be borne out in human clinical trials, and we're getting there as quickly as we can. There are several steps that we have to undertake in order to get this drug into human clinical trials. Right now, we are conducting dose ranging studies to better understand at what dosing level we have to deliver the drug to see an effect. We'll be scheduling a pre-IND meeting with the FDA in the coming months in order to introduce this program to them, help build some familiarity with the compound and what we're trying to accomplish, and get some feedback in terms of what our proposed pathway is to get into human clinical trials. We also have what's called GLP studies. These are toxicology studies and other studies that are required prior to treating humans. And so it's a somewhat of a long process, an expensive process, but we're committed to going down that path with this compound. At the same time, we continue to seek co-development partners and strategic investors to bring in the funds necessary to help accelerate the development of the INM-901 program in Alzheimer's. So thank you very much for listening to this presentation today. If you have any further questions about the company or about the programs, please feel free to reach out to Colin Clancy at the information listed here, and we'll be glad to get back to you. Thanks again.